Good morning, anatomy students! So, today we are going to be going through the muscular system. So, this is going to be one of the 11 systems of your body. It is going to contain your muscles. So, we are going to start with the functions of your muscular system. It converts chemical energy into kinetic energy. It converts stored energy in chemical bonds into energy of motion, kinetic energy. It provides muscle tone, it moves fluids and food, it generates your heartbeat, it produces and distributes heat. So your muscles are incredibly important to your function. So there are 3 types of muscle tissue. We have talked about these before when we were going through tissues. They are skeletal muscle, smooth muscle, and cardiac muscle. If I just say muscle tissue, I tend to mean skeletal muscle. That is the muscle of your named large muscles. So most of the time, I'm going to be talking about skeletal. So skeletal muscle is almost always going to be attached to bones. It is voluntary, which means you control it consciously. And it is striated. Striated means it looks striped. So you've seen this under a microscope. This is blown up probably near 2,500 times. I think you guys saw these little stripes though. So it is going to be striped, attached to muscle, voluntary. Smooth muscle is going to be the walls of organs, of viscera. You also have it for your erector pili muscles for your hair. Your smooth muscle is involuntary. It is not under your conscious control. You cannot think and make your hair stand up on end. It'd be cool, but we can't. It is non-striated. It does not look stripy under a microscope. It has much shorter cells. So your skeletal muscle cells will run the whole length of the muscle. These are much shorter and do not. And there's cardiac muscle. Wall of your heart. It is involuntary. It is striated. It has the intercalcated discs, which differentiate it, and it's got this branching pattern that we don't see with skeletal muscle. So we're going to move on to your muscular system. I forgot I put that joke about steak doneness on here. Well, it's not a joke. That is different degrees of steak doneness, but I forgot I included that on here. But I did, I guess. So muscles are individual organs. You have around 320 pairs of muscles. So convenient thing is we are bilaterally symmetrical. So you have the same muscles on the right side of your body that you do on the left side of your body. In the past, I have made students responsible for memorizing these muscles. Now, with our current at-home status, I don't know how that quiz will work. I certainly will be assigning coloring. I might try to do a lab where you have to figure out the names like I did for tissue ID and for the bones. That might be more difficult for this. I'm not sure. Tentatively, I'm going to say that you should learn these muscles. So there are 27 on the front here. There are 25 on the back. These are all on page 314 in your text. You have this exact picture on page 314. So please take a look there. These are going to be your largest muscles. So now now we're going to go through the anatomy of skeletal muscles. So, muscles are surrounded by dense connective tissue called fascia. Fascia becomes tendon when it attaches to bone. So, muscle is covered with connective tissue. If you ever, I'll talk about that if I had you in person. I'm not recording that. So, fascia is going to become tendon when it attaches to bone. Some fascia forms broad sheets, which are epicondroses, which you have here. So you have these sheets of connective tissue of fascia. So the tissues of skeletal muscle there's going to be skeletal muscle tissue. There's also nervous tissue. Your muscles are innervated. They must have nervous tissue to tell the muscles what to do. There's going to be blood, and then there's going to be various connective tissues. So it is not just skeletal muscle. There's going to be nervous tissue, blood, and connective tissues there as well. Now, we're going to get into the organization of skeletal muscle. Skeletal muscle is organized in bundles, essentially. So, you have a muscle. Muscles are made out of groups called fascicles. So a fascicle is this bundle, so it looks like this muscle here has 1, 2, 3, 4, 5, 6, maybe another 7th fascicle hidden back there. Then the fascicles themselves are made of muscle fibers. So here we have a muscle fiber. The muscle fibers are the cells. So what we call a muscle fiber is a muscle cell. It's actually a whole bunch of cells that are sort of fused together, but it's continuous. There's no break in its cytoplasm. So there are layers of connective tissue that go around each one of these things. So there is epimysium surrounds the whole muscle. Perimysium divides the muscle into fascicles. The fascicles are divided into muscle fibers. Then the muscle fibers are surrounded by endomysium. So there are 3 different types of connective tissue layers that are doing surrounding. Epimysium is surrounding the whole muscle. Perimysium is surrounding a fascicle. And then we have endomysium is surrounding a muscle cell. So now we're going to go through the organization of muscle fibers. So again, the muscle fibers are the cells. So these are single cells. They're formed from many fused cells, so they have many nuclei. So this is an exception to our all cells have one nucleus thing, because these cells have a whole bunch of nuclei because they fused. They are centimeters long, but that little U is micrometers in diameter. Like your skeletal muscle fibers can span like your whole length of like your muscle. They are incredibly long for a cell, but they're incredibly thin. The cell membrane is called a sarcolemma. The cytoplasm is called sarcoplasm. They have a ton of mitochondria in them because mitochondria are the powerhouse of the cell, and it needs lots of energy. And myofibrils are the contractile units. So, within a muscle fiber, there are these tubular groups of protein called myofibrils. They then are broken into sarcomeres. So, now we're going to talk about the organization of a myofibril. This is a group of protein. It's made of thick filaments, myosin, and thin filaments, actin, as well as troponin and tropomyosin. So there are 4 proteins in these myofibrils. So myofibril, so this whole thing is the muscle cell. So we got a nucleus, and it's this long, skinny cell. Here we got another nucleus, big, long muscle cell. It'll be centimeters long. Very skinny though. Then these bundles that are in it are a myofibril. Those bundles are made of protein. They're made of just 4 proteins. They are made of thick filaments, which are made of myosin. So this pink here is myosin. Then there are these purples. So the light purpley dots are going to be actin. Then there is going to be troponin, and tropomyosin are these dark purples. So this is a very basic diagram showing a sarcomere. So sarcomeres have all of these parts. So skip up to where we actually start talking about them. I might skip back to the slides in a bit. So sarcomeres are what give muscle its striped appearance. So its thin and thick filaments create striation patterns, which are stripes. The unit of thick and thin filaments is called the sarcomere. So the stripiness that you saw with your eyes looking through the microscope is being caused by the microscopic arrangement of these proteins within the muscle fibers. And all of these proteins are arranged, aligned in the same way, which is why you can see this under microscope. So sarcomeres have a few different parts. So there are eye bands. Eye bands are going to appear light under microscope. So this is now going to be a very powerful microscope that is showing this. I don't think this is electron microscope imagery, but it could be. I'm not actually sure. So the eye band is going to just have the thin filaments. So that just has the actin filaments in this eye band. Then there is the A band. The A band is going to be dark. That is where there is thick filament. Then there is the H zone. In the H zone, that is where it's a bit lighter. We only have the thick filament, so since there's no overlap with the thin filament, it's a bit lighter there. Then there's the Z lines. The Z lines are so named because these actins, they attach in this Z pattern. So it cuts eye bands in half with a zigzag. So that is why it is a Z line. Then we have a M line right down the middle. So the M line cuts the center of the H zone in half. So when a muscle contracts, what happens is these thin filaments and thick filaments pull together like you're pulling your fingers together, and when your muscle relaxes, they pull back apart. And when you have a large skeletal muscle that's contracting, you just have thousands and thousands of these units that are pulling together a little bit to create that motion. So, the thin filaments are actin proteins. They are helical in shape. Troponin and tropomyosin are proteins that bind to the actin to make it inaccessible to the myosin. So the tropomyosin is this purple, also helical protein. Then the troponin is this bubblegum pink thing. Now, when calcium ions interact with troponin, it pulls the tropomyosin away from all these little black marks on this actin are the active sites. And if calcium comes in, that exposes the active sites, and that allows myosin to engage on the actin's active sites. So, there are a few parts of a muscle fiber we need to talk about. So, all of the organelles of muscle fibers are highly specialized. So there are a lot of organelles that have their own special name when it's in a muscle cell. It's doing all of the main functions that it does in our protocell that we learn about that doesn't actually exist. But since the functions have become so much more specialized in this tissue, they have different functions and names. So there's sarcoplasmic reticulum, which is a fancy endoplasmic reticulum. There are transverse tubules, these are cellular invagination, so these are going to go in through the cell. So if you see those little pores are then going to penetrate in through the cell. The sarcoplasmic reticulum for Cristaceae, that is these groups which form a triad. So there's two cristaceae and a transverse tubule going through. So there's all of these passageways formed from endoplasmic reticulum and from the transverse tubules. So I think I'm going to stop here, but to go through what we've talked about so far. So we're mostly talking about skeletal muscle right now. There are the two other types. So skeletal muscle is going to be attached to bone by tendon. Skeletal muscle is going to be formed of groups called fascicles. Those fascicles will be formed of muscle cells. But muscle cells are going to be formed of myofibrils. The myofibrils have a functional unit of the sarcomere. The sarcomere has this layout where there are thin filaments and thick filaments, and those thick filaments and thin filaments can slide past each other to contract. The thick filaments are made of myosin. The thin filaments are made of actin. They have tropominin and tropomyosin that are associated with them. So if you got it this far, I hope I've not totally bamboozled you. I am going to be posting a few anatomy crash course videos that I need you to watch along with this because I need you to do that. So, have a good day.